For nearly 190 years, every image you've ever seen of Megalodon, every documentary, every museum exhibit, every size comparison chart, was built on the same assumption. And that assumption was never actually proved by the evidence. Here's what we got wrong, and how one fossil in Belgium changed it. The problem begins with biology. Sharks belong to a class called Chondrichthyes, the cartilaginous fishes. Their skeletons contain no bone at all. What holds a shark together is cartilage, the same flexible tissue you have in your ears and nose, stiffened only by a thin outer crust of mineral deposits. When a shark dies and sinks to the seafloor, the soft tissue rots quickly. The cartilage follows. Without anything hard enough to withstand millions of years of sediment pressure and chemical change, the skeleton simply dissolves back into the water it came from. What survives is the teeth. Shark teeth are mineralized with fluoroapatite, a calcium phosphate compound so durable it can outlast geological ages without degrading. A single shark produces tens of thousands of teeth across its lifetime as it continuously replaces them, and those teeth rain down through the ocean floor and into sediment in extraordinary numbers. For Megalodon specifically, the teeth can exceed 7 inches in length. They are hard, dense, and everywhere in the fossil record. The skeleton is nowhere. There is one partial exception. The vertebral centra, the disc-shaped cores of the vertebrae, are composed of what paleontologists call calcified cartilage, a denser, more mineralized tissue than ordinary cartilage. Under specific conditions, rapid burial, sediment chemistry that favors mineralization, vertebral centra can fossilize. Jason Bonnan, professor of biology at Stockton University and a co-author on foundational vertebral studies, describes the distinction precisely. Shark vertebrae are made up of calcified cartilage, not the rubbery cartilage at the end of your nose or in your ears, but a special type of cartilage that has minerals in it, making it more likely to be preserved as a fossil. That's why we have fossils of megalodons at all. But preservation is the exception, not the rule. For Megalodon specifically, researchers have identified only 2 known vertebral column specimens in the entire fossil record, one from Belgium, one from Denmark. Every other locality that has ever produced Megalodon teeth has produced no vertebrae at all. A 2022 study in Science Advances states this directly. Vertebral columns of extinct sharks hardly ever preserve. Imagine an X-ray of a beaver's tail. The image reveals only a delicate spine threading through the tissue. The wide, flat, paddle-shaped structure visible on any living beaver is pure, soft tissue, no skeletal trace of it whatsoever. From the hard parts alone, you would reconstruct something unrecognizable, and you would have no way of knowing what you'd missed. That is precisely the position Megalodon science has occupied since the first tooth was formally described. The body shape, the proportions, the girth, the fins, everything beyond the mineralized fragments had to be inferred from other animals entirely. For 190 years, that inference ran through the great white shark. October 1666. 2 fishermen working the Tyrrhenian Sea between the islands of Gorgona and Meloria off the Tuscan coast drag up something enormous. The animal is so large and so violent that it can't simply be hauled aboard. It must be restrained and bludgeoned into submission after, according to the account, being lashed to a tree. A letter written by the philosopher Lorenzo Magalotti on 26 October 1666 records the capture in remarkable detail. The creature's liver alone weighs approximately 300 pounds, its other viscera around 2,400 pounds, the full animal estimated at roughly 3,500 pounds. The head is separated from the body and transported by road to Florence, where it's presented to the Danish anatomist Nicolaus Steno, serving at the time as physician to the Medici court under Grand Duke Ferdinando II. The dissection isn't a private affair. Florence in 1666 is a city of natural philosophy and court spectacle, and a severed shark head the size of a cartwheel arriving from the Tuscan coast with its jaw still intact draws an audience. Steno works in front of them. He peels back the tissue, counts the tooth rows, 13 in the upper jaw, 13 in the lower, and lays the teeth out where they can be examined. Then he reaches for something that would have been familiar to almost anyone in the room, a glossopetrae, one of the triangular stone objects found in abundance on the shores of Malta and across the European coastline. Christian tradition had made these objects famous. Pliny the Elder had described similar stones as having fallen from the sky during lunar eclipses. Medieval and Renaissance folklore identified them as the petrified tongues of serpents, turned to stone by the Apostle Paul on Malta, hence glossopetrae, tongue stones. They were sold as amulets, sewn into the hems of garments, embedded in reliquaries, and worn as protection against poison. Steno holds one beside the shark's freshly dissected tooth, and the shape is unmistakable. Same triangular form, same serrated edge, same root architecture. Just stone rather than enamel. He publishes his findings in 1667, not 1666 but the following year, concluding that those who assert that large tongue stones are the teeth of a shark aren't far from the truth. An Italian naturalist named Fabio Colonna had already argued the same identification in a publication around 1616. Steno's contribution was less the identification itself and more the geological synthesis that followed, formalized in his 1669 treatise De Solido, which established foundational principles of stratigraphy and fossil formation. But what the 1667 shark head work demonstrated was a method, inference from a living analogue to an extinct specimen. He had a real shark in front of him. Its teeth matched the fossils. Therefore, the fossils were teeth. The logic worked because the analogue was correct. That same logic, use the living animal as the key to the extinct one, would carry forward into every subsequent attempt to understand Megalodon. The method wasn't flawed in principle. The flaw came when the method outran the evidence available to support its conclusions. In 1835, the Swiss naturalist Louis Agassiz was midway through his monumental Recherches sur les poissons fossiles, a multi-volume taxonomy of fossil fish that would eventually describe over 1,700 species. In June of that year, the fifth installment of his atlas included Plate 29 of Volume 3, illustrating a set of large triangular fossil teeth under the label Cartilaginous Megalodon, with a brief insert noting, "Large fossil species of Cartilaginous to which I give the name of sea Megalodon." This is the moment the name Megalodon enters the scientific record, and the official date under the International Code of Zoological Nomenclature is 1835. 8 years later, in the 1843 text volume of Recherches, Agassiz formally described the species and reassigned it. The genus Carcharodon, containing the great white shark, had been established by Müller and Henle in 1838. By 1843, Agassiz had moved Megalodon into it. Carcharodon megalodon, in the same genus as the largest living predatory shark. The reasoning was defensible. Megalodon teeth and great white teeth share the same gross morphology, large, triangular, serrated crowns. The great white was the most ecologically comparable living shark. Agassiz was working from the best comparative data available, and the limitation wasn't his rigor. It was the evidence itself. He had no skeleton to examine, no body proportions to measure, no structure beyond the teeth that could tell him anything about how the animal was built. So he drew the only inference the evidence permitted. The teeth resembled the great whites. The great white was the largest predatory shark alive, and if the fossil matched the living animal closely enough, the living animal was the appropriate model. What Agassiz didn't and couldn't do was reconstruct a body. His treatment was strictly taxonomic: teeth, classification, genus placement. He drew no skeleton, proposed no proportions, described no behavior. But the genus placement carried an implication that would persist for over 160 years. Megalodon belonged to Carcharodon. Therefore, the great white was its closest living relative. Therefore, the great white's body plan was the appropriate template. No fossil skeleton existed to challenge this, so the implication filled the gap. It would fill that gap for generations. In 1996, Michael Gottfried, Leonard Compagno, and S. Curtis Bowman published a chapter in the edited volume Great White Sharks, The Biology of Carcharodon carcharius, that would define the public image of Megalodon for 3 decades. Their paper, Size and Skeletal Anatomy of the Giant Megatooth Shark, Carcharodon Megalodon, took the great white comparison from a taxonomic placement to a physical object. The methodology was made explicit. They regressed the great white shark's total body length against the height of its largest upper anterior tooth, then applied that regression to Megalodon teeth. A great white skeleton was scaled up accordingly, modified for a broader cranium, shorter snout, more massive jaws, and proportionally larger pectoral fins. The result was a maximum total length estimate of approximately 15.9 meters and a body mass of around 48 metric tons. Gottfried and colleagues had to rely heavily, in their own framing, on extrapolations from the modern great white shark, which they believed to be its closest living relative. The result was a reconstruction the Bristol Dinosaur Project would later summarize plainly. A skeletal reconstruction led by Mike Gottfried in 1996 subsequently gave it the look of a stockier great white. It made sense at the time. From that model, the visual standard for Megalodon propagated outward into physical public space in a way that no academic paper can fully achieve on its own. The American Museum of Natural History in New York had built its first Megalodon jaw reconstruction as early as 1909. The Field Museum in Chicago mounted a jaw display 50 feet across. The Smithsonian National Museum of Natural History featured the animal in its permanent collection. Documentaries adopted the silhouette. Educational materials adopted the size comparisons. What mattered about the 1996 model wasn't just that it was published, but that it became walkable. A person could stand in front of it. A child could put their head inside the jaw replica and have their photograph taken. The reconstruction gave the inference a texture and a scale that a paper never could. The rough fiberglass surface, the rows of cast teeth arranged in their precise dental formula, the swept-back pectoral fins positioned exactly as they would be on a great white, only larger. Docents described what they were showing visitors as a reconstruction based on the best available evidence. That description was accurate. The evidence was teeth and the analogy that teeth had suggested. The exhibit didn't advertise what it lacked, the animal's actual structure. Why would it? Nothing in the room suggested there was anything to doubt. Across all of these venues, the visual vocabulary was consistent. A vast, barrel-chested predator, an amplified great white, its outline stamped onto every museum wall and television screen. What visitors were looking at wasn't a discovered animal. It was the original inference. Agassiz' 1843 taxonomic comparison translated into fiberglass, steel, and illustration. The assumption had become a cultural object, and cultural objects are harder to revise than scientific papers. The first scientific challenge came not from a new skeleton, but from closer examination of the teeth. From the late 1990s through the 2000s, researchers studying tooth morphology in detail found differences between Megalodon and the great white that were more substantive than the gross similarity had allowed. The teeth of the two animals diverge in several measurable ways. Megalodon teeth carry a distinctive dark enamel band at the base of the crown, a feature called a bourlette, that is absent in great white teeth. The root structures differ substantially. Megalodon roots are thick and heavily curved, forming a broad foundation, whereas great white roots are narrower and lighter. The serration patterns differ in density and arrangement. Fossil evidence also pointed the great white's actual evolutionary ancestry toward mako-like sharks, Isurus and related forms, rather than toward the megatooth lineage. The dental resemblance between Megalodon and the great white appeared on closer analysis to be convergent evolution. Two unrelated lineages developing similar teeth because they occupied similar ecological roles, not because they shared recent common ancestry. The taxonomic revision was gradual. Henri Capetta formalized Otodontidae as a distinct lamniform family in his major 2012 synthesis of fossil shark taxonomy. Kenshu Shimada and colleagues formalized the genus framework in a 2017 paper in Historical Biology. By the time the reclassification was complete, the animal's name had become Otodus Megalodon, placed within an entirely extinct family whose lineage traces back through a series of Otodus species to Cretaceous predecessors. The genus Carcharodon and the direct relationship to the great white it implied was gone. The Natural History Museum in London stated the situation directly. Most reconstructions show Megalodon looking like an enormous great white shark, Carcharodon carcharias, and this is now believed to be incorrect. But the correction had a structural limit. Moving Megalodon into Otodontidae changed its evolutionary address without changing its picture. No alternative skeleton existed to replace the great white template. Body plan reconstructions continued using lamnid analogues because there was nothing else hard enough to work from. Science had corrected the family tree. The drawn animal in the museums barely shifted. The story of how that second correction finally arrived begins in a museum basement in Brussels, with 141 vertebral centra that had been sitting there since the 19th century. The specimen designated IRSNB P9893 was collected near Antwerp from Miocene deposits, the Sand of Antwerp Formation, and deposited at the Royal Belgian Institute of Natural Sciences in Brussels. Its composition: 141 associated vertebral centra from a single individual, the largest centrum reaching 15.5 centimeters in diameter. A researcher named Lariche studied it in 1926, published briefly on it, and the scientific community moved on. For most of the 20th century, as museum reconstructions were built and documentaries commissioned and exhibitions mounted, the Belgian specimen sat in storage in Brussels, in a drawer alongside a collection of isolated teeth found near the same sediment layer. The teeth and the vertebrae weren't necessarily from the same animal. The association wasn't certain enough to claim they were. What was certain was the scale. Vertebral centra 15.5 centimeters across, arranged in a sequence suggesting a trunk of remarkable length, from a Miocene deposit known to contain Megalodon material. Its significance became clear when researchers applied modern imaging. The process wasn't dramatic in the way excavations can be. There was no dig, no discovery moment, no field camp. The centra were already in Brussels. Shimada, working during the pandemic restrictions of 2020 and 2021, coordinated with Bonan remotely. The Belgian specimens were loaded into a CT scanner at the Royal Belgian Institute, and the resulting files were transmitted electronically to researchers in New Jersey and Chicago. Bonan received the data and began building virtual cross-sections from the X-ray slices, a three-dimensional reconstruction of the vertebral interior, revealing the concentric growth rings that record each year of the animal's life, the way tree rings record drought and rainfall. That work was published in 2021. The 2022 study in Science Advances, with a broader international team, CT scanned and three-dimensionally modeled the entire precaudal vertebral series. The result? A trunk length of approximately 11.1 meters. That is the spine alone, measured, not inferred, from fossil material in a museum in Brussels. The AMNH blog described it simply: the animal's spine alone was about 36 feet, 11.1 meters long. The University of Pisa's summary put the scale in plain terms. Scientists had analyzed an extraordinary fossil, a partial vertebral column consisting of 141 vertebrae, approximately 11 meters long, found in Belgium. The 2022 study estimated the Belgian individual's total body length at around 16.4 meters using this vertebral data. But that number wasn't the most consequential finding from the specimen. The most consequential finding was what it revealed when placed beside the previous model's estimate for the same animal. Gottfried and colleagues in 1996, using their great white scaling regression, had estimated the same Belgian individual at approximately 9.2 meters total body length. A 2024 paper by Stearns, Shimada and colleagues, published in Paleontologia Electronica, identified the problem directly. The total length estimate was shorter than the measured length of the spine alone. 9.2 meters total, including head and tail, for an animal whose trunk vertebrae alone measured 11.1 meters. The arithmetic wasn't imprecise. It was impossible. Hold that for a moment. The most widely cited, most widely reproduced scaling methodology in Megalodon science, the one that generated the reconstruction in the museum, the one that produced the 15.9-meter estimate that anchored a generation of educational materials, produced, when tested against the one specimen where it could be checked against actual measured structural data, a total body length shorter than the measured spine. The Great White scaling method wasn't merely inaccurate. Applied to the specific evidence available, it was geometrically incoherent. It had been geometrically incoherent for decades. No one had been able to demonstrate this until the Belgian vertebral series was properly measured and modeled. In March 2025, Shimada, professor of biological sciences and environmental science at DePaul University in Chicago, led the publication of a comprehensive reassessment of Megalodon's biology in Paleontologia Electronica, DOI 10.26879-1502, with 28 named co-authors from institutions across the United States, Europe, Japan, and Australia. The paper's title set out its scope: a reassessment of the possible size, form, weight, cruising speed, and growth parameters of Otodus megalodon, alongside new evolutionary insights into its gigantism, life history, and extinction. The method was a departure from anything attempted before. Rather than selecting a single modern shark as the scaling template, the team surveyed the proportional relationships between trunk length, head length, and tail length across a broad sample of living and extinct Neoselacian sharks spanning multiple orders. They used the Belgian vertebral column as the fixed structural anchor, a measured trunk length rather than an inferred total length. Then they applied the median proportions from the broad comparative survey to calculate what the head and tail would have contributed. The upper range total body length estimate that emerged was approximately 24.3 meters, close to 80 feet. For scale, a sperm whale typically reaches around 18 to 20 meters in length. But the 24.3-meter figure is derived not from the Belgian specimen directly, but from a second specimen. Enormous vertebral centra from the Gram Formation in Denmark, up to 23 centimeters in diameter, which the 2025 paper used to extrapolate the maximum plausible individual size. Those Danish vertebrae had originally been excavated in 1983 by Bendix Almgreen, studied briefly, then became difficult to locate physically for decades. Their existence documented only in photographs in the scientific literature until their recovery. The Belgian individual, on the new methodology, comes out at around 16.4 meters. Shimada stated the 24.3-meter figure with appropriate precision. The length of 24.3 meters is currently the largest possible scientifically justifiable estimate for O. megalodon. Not a confirmed measurement, but a scientifically justifiable maximum, derived from the largest confirmed vertebrae and applied through the most rigorous comparative framework yet attempted. The number generated headlines. The shape mattered more. The Belgian vertebral column, examined comparatively across the dataset, was, in the study's own language, remarkably slender relative to white shark vertebral columns of comparable inferred body size. The vertebral centra were proportionally thinner than a great white's at the same implied length. The 2025 study's hydrodynamic analysis went further. Scaling up a great white body plan to 16+ meters would produce an animal with excessive drag, a body too broad, with too much frontal area for efficient locomotion at that size. The physics favored something longer and leaner. Stearns, who contributed to both the 2024 shape paper and the 2025 comprehensive study, described the finding in commentary for UCR News. Rather than resembling an oversized great white shark, Megalodon was more like an enormous lemon shark, slender and elongated in body form. That shape makes considerably more sense for moving efficiently through water at that scale. Shimada told National Geographic the same point from the other direction. Previous studies simply assumed that Megalodon must have looked like a gigantic version of the modern great white shark, without any evidence. One correction to 190 years of image making. Arrived late, built on calcified cartilage from a Belgian basement. The shape change carries behavioral implications that reach into every piece of Megalodon content you've encountered. The difference in body plan isn't subtle. A great white shark has a broad, torpedo-shaped midsection that tapers sharply toward the tail. A compact, heavily muscled form built for explosive acceleration from a relatively short body length. Place a great white beside a lemon shark, and the contrast is immediate. The lemon shark is longer relative to its girth, more cylindrical through the trunk, with a less dramatic taper. The proportions are closer to a large, sleek cruiser than to a missile. Scale either body plan up to Megalodon's estimated length, and you get radically different pictures. The stocky great white scaled version fills the frame. The lemon shark proportioned version stretches across it. Museum sequences and documentary footage have depicted Megalodon in the great white mode, rising from depth, attacking from below, overwhelming prey in a single catastrophic strike. That image came directly from the great white template, and it has been reproduced so consistently that it functions as common knowledge. If the body was structurally different, longer, leaner, less suited to explosive burst speed, the predatory mechanics were likely different too. An elongated, more cylindrical body plan is associated, across large marine animals generally, with sustained cruising efficiency rather than burst speed ambush. Tim Hyam, a UC Riverside biologist and co-author of the 2025 paper, articulated the underlying principle. Evolution moves toward efficiency, and a body built for sustained locomotion reflects that. The 2025 study estimated a cruising speed for a 24.3-meter individual at approximately 2.1 to 3.5 kilometers per hour. Slow in absolute terms, entirely appropriate for an animal managing the caloric demands of extraordinary size across open ocean. The behavioral inference is exactly that, an inference. No direct record of how Megalodon hunted exists. What we have are bite marks on fossil whale bones, documented at sites including the Pisco Formation in Peru and the Calvert Cliffs of Maryland, showing Megalodon targeted cetaceans and attacked from multiple angles. Whether those attacks came as explosive ambushes or sustained pursuits is precisely the kind of detail that teeth and bite marks can't settle. What the corrected body plan suggests is a different default mode from the great white. An ambush predator and a sustained cruiser aren't cosmetically different. They represent different strategies for locating prey, different energetic demands, different relationships to the ocean environments they inhabited. The entire behavioral model the audience carried from decades of documentaries was sourced from the wrong animal. The question of whether Megalodon could still exist has a cleaner answer than the body shape question, and the evidence is considerably firmer. Megalodon's teeth disappear from the fossil record at approximately 3.6 million years ago, during the early Pliocene. This consensus date comes from a 2019 study by Robert Bersenecker and colleagues that filtered out reworked specimens, teeth displaced from older sediment layers into younger ones, which had previously inflated survival estimates. After that filtering, the last reliable Megalodon occurrence sits at 3.58 to 3.6 million years ago. The species had existed for roughly 20 million years. The causes of its disappearance were multiple and reinforcing. Ocean cooling during the Pliocene reduced the warm coastal shelf environments where Megalodon fed and reproduced. A 2016 analysis by Catalina Pimiento and colleagues found that Megalodon's geographical range began contracting in the late Miocene, coinciding primarily with the decline in diversity of filter-feeding whale species and the emergence of new competitors, early killer whales and the expanding great white shark. Some accounts propose Megalodon simply evolved into a smaller form as conditions shifted. The fossil record provides no support for this. Evolution into a smaller form would produce transitional fossils, animals with progressively smaller teeth in progressively younger sediment layers. There are none. Megalodon teeth disappear abruptly from the stratigraphy at 3.6 million years ago. There is no graduated transition. There is absence. As for survival claims, the footage misidentified as Megalodon, the deep-sea speculation, the argument collapses on basic ecology. In 2013, the Discovery Channel broadcast footage it claimed showed a living Megalodon, footage that was fabricated for a mockumentary. That same year, a large great white shark tagged off South Africa disappeared from tracking data at extraordinary depth, briefly generating speculation online. Both episodes resolved to nothing. Stephen Godfrey, curator of paleontology at the Calvert Marine Museum in Maryland, makes the point plainly. If Megalodon was still alive today, people wouldn't be wondering if they saw one. They would know. It's hard to hide an 80-foot-long predator that has to live where its prey are. The whales and dolphins that Megalodon required for sustenance breathe at the ocean surface. A predator of that metabolic demand can't feed in the deep ocean and can't conceal itself while feeding in shallow water. Every claimed sighting has resolved on examination to misidentified species or fabricated footage. No physical evidence of Megalodon predation on modern marine mammals has been found anywhere in the world. The animal is gone. The question is, what we built in its absence. Return to the Belgian specimen. IRSNB P9893 was collected in the 19th century, studied intermittently through the 20th, CT scanned and modeled in the 21st, and published as the structural anchor of a comprehensive new analysis in March 2025. Its vertebral column, 141 disarticulated centra spanning approximately 11.1 meters, sat in a museum in Brussels for more than a century, while every documentary, every exhibit, every comparative chart proceeded on the basis of teeth from other localities. The teeth were more numerous, more globally distributed, easier to analyze. The vertebrae required specialized facilities, international collaboration, and modeling software that didn't exist for most of the specimen's institutional life. The 2025 Shimada study drew on a broad comparative survey of shark species across multiple orders. It had 28 named co-authors from institutions across 4 continents. Its methodology was explicitly designed to avoid the single analog problem that had constrained every previous reconstruction. And it arrived 190 years after Agassiz named the species from a handful of teeth. In those 190 years, what changed wasn't the available evidence. Megalodon teeth had been collected globally since the 17th century, but the type of evidence being used. Every size estimate, every body plan reconstruction, every behavioral inference had been derived from the same class of hard parts, the teeth. The vertebral column data had been available in principle since Lariche studied the Belgian specimen in 1926. But without a methodological framework that could use it independently of the Great White template, it couldn't break the model. The 2024 Stearns, Shimada et al. Shape paper, followed by the 2025 Comprehensive Reassessment, was the first systematic analysis to demonstrate that vertebral structural data and tooth scaling methodology produced materially different results, and to use that discrepancy as the argument for replacing the older approach. Jack Cooper, a paleobiologist at Swansea University who wasn't part of the 2025 study, acknowledged both the advance and its limits in commentary for National Geographic. Could Megalodon have been slenderer than previous reconstructions? Sure, it's possible, but the study doesn't outright say how much slenderer, nor does it definitively rule out previous interpretations. Shimada and his co-authors are similarly measured in the paper itself, describing their conclusions as working hypotheses expected to serve as reasonable reference points for future studies. A complete skeleton would confirm or revise them. No complete skeleton exists. None has ever been found. The shape we built for Megalodon was the shape of the nearest available analog, not the shape of the animal itself. Steno proved the comparative method valid in 1667. Match the teeth to the living analogue, and you can identify the fossil. For Megalodon, the method broke down not because it was wrong in principle, but because the living analogue was wrong. Agassiz, in 1843, had no way of knowing that Megalodon's lineage had diverged from the great whites tens of millions of years earlier than the tooth similarity implied. Gottfried, in 1996, was building on the best available framework. His paper said as much, that the great white was believed to be the closest living relative. The reclassification into Otodontidae in the 2000s corrected the family tree, but couldn't update the picture without structural data to replace what had been removed. Each generation of researchers worked honestly from what the evidence offered. The problem was architectural. A cartilaginous animal leaves teeth far more reliably than it leaves anything else, and the science that grew from teeth calcified around assumptions that couldn't be tested until the skeleton arrived. One spine, one vertebral column, 141 disarticulated centra, collected in the 19th century and deployed properly in the 21st. That single specimen exposed a logical impossibility in the previous scaling model, pointed toward a longer and leaner animal than 190 years of illustrations had shown, and suggested a predatory mode that diverges significantly from everything the great white template implied. Whether the 24.3-meter upper estimate holds, whether the lemon shark analogy survives as more vertebral specimens are recovered, whether the cruising behavior inference will find further support, all of that remains open. The 2025 paper is explicitly a set of working hypotheses, not a final settlement. Science has been here before with Megalodon, a confident model standing until a different type of evidence forces a reckoning. The honest answer is that the reckoning is incomplete. We have corrected the size and begun correcting the shape. The full anatomy, the soft tissue distribution, the fin geometry, the coloration, all of it remains, in the strictest sense, unknown. The fossil record will never deliver a complete Megalodon. The gaps we filled with the great white analogy will need to be refilled with something else, and the something else will itself be inference, constrained by whatever hard parts the ocean floor eventually yields. What isn't working hypothesis is the structural problem that made those hypotheses necessary. The reconstruction failure was real. It was baked in at the origin. Every image you have seen of Megalodon was assembled from fragments, filtered through an assumed relationship, and presented as though it were a portrait of the actual animal. We weren't discovering Megalodon for 190 years. We were constructing it from the only pieces hard enough to last. And it turns out we built it wrong. Subscribe for more stories like this.